So, so despite my amazing success at introducing the last session, I am now also going to introduce our first plenary speaker. So Phil Hodgkin is the head of the Immunology Division at Walter and Eliza Hall Institute for Medical Research in Melbourne. And you know, there's a joke that Bertrand Russell once said that if you meet a beautiful woman at a party, you should also always tell her that she's one of the two most beautiful women that you've ever met, in case that another woman that you said to that before was standing behind her, and so that way nobody will get offended. Okay, so I have to say that Phil Hodgkin was one of the, and we have another plenary speaker, so he's one of the three people I first considered when I thought of who to invite as, you know, a plenary speaker for this session. And really, I mean, we talk a lot about, oh, not we, I don't know about we, but I talk a lot about the need to do interdisciplinary, multidisciplinary biology. And I've been talking about it for a long time, since I started trying to do it myself. And always my example as a person who actually does everything that usually you need a whole collaboration set of people doing, it's Phil. So, and today and I had another interesting story about him, but we don't have time, so I'll tell it to you later over the meal. And so today I think he's going to talk about, well, I don't know exactly because I haven't heard the talk, but again, look at Stochastic immunity, more than the repertoire. So,
well, thank you, Uri. Thank you to all the organisers for the fantastic invitation to speak here. I was here at the first one that Ramit organised, and it was fantastic to see a repeat and to see that this theme is growing. I think we are part of something very special in immunology, the role of stochasticity. I think the story Uri was going to tell us that I cancelled my mother's 80th birthday in order to make it here, because I thought any conference about stochasticity and immunity, I had to be there. So thank you for that. My mother sends her regards.
have a wonderful present waiting for that.
That's right. So just to give some context, this is my theme for the day. How do we start off with a cell, generate and cells capable of protecting us in some immune response? So the two steps that we usually think of for the immune response are building the repertoire, and we've heard already a lot about that, and we'll hear more in this conference. And the second step is really to challenge that repertoire and watch it undergo complex fate changes and control. So in that first step, of course, we make a perceptive cell. We know this goes through some stochastic process here, and I'm not going to draw the picture of the molecular rearrangement, although I could have at this point. Other than just to say, of course, there's no question this is a stochastic process. And the features therein are that we have amazing coding efficiency. With just a few hundred genes, we can encode millions and billions of alternative sequences. The other feature is that because of this cells going through some stochastic process here, the final cell here is indeterminate. We can't look at this cell and predict what it will end up here in terms of sequence. And that in order for the system to operate, we have to work with large numbers. So the single cell can't mount an effective immune response against all the challenges. Okay. Now the other point I want to make is that perceptive cell, in addition to the receptor, the antigen receptor, whether T or B, has many other receptors on its surface, many other ligands. This is from a review some years ago showing that there's about 60 other cell surface molecules on that cell. Each of those molecules, if you looked at it by flow cytometry and measured individual levels on the population, you would see it would follow a distribution, usually a log normal distribution, quite broad. So here's, in addition to the antigen receptors, we've got at least 60 other receptors that are distributed on the surface of those cells, independently of the receptor. And when you measure them against each other, very often they're independent of each other. So they also have some sort of independent control. The consequence of this is that if you drill down carefully, you conclude that every cell is unique and therefore has the potential to perceive its environment differently from any other cell. So we have generated our probabilistic cell. Our final cell, I would say, is a symphony of randomness, not just the receptor, but all the other components in the cell, on the cell surface and internally. We can treat this as noise and work from means, which is a traditional view of how to deal with that diversity. But there's a more interesting possibility, which is that variations in behaviour can emerge from this ensemble of different perceptions based on the stochastic construction of the cells. And I'm going to look for evidence of this as I work my way through some experiments looking at step 2 in the immune response. So we've generated our cells. Now we want to challenge them and watch them undergo complex fate changes. And of course, this shown here, the proliferation, cessation, death leading to memory. The colours indicate the diversity of cell populations that are being generated along the way. And we can think of all these other cell types that are being generated as the second repertoire, the functional repertoire that the immune system has to deal with. Now, how complex is that subsequent behaviour here? It turns out to be really complex. So we can have many different challenges to this immune response. Viruses, bacteria, we have to deal with self. We have parasites, we have toxins. We have many sentinel cells contributing to the information that the T and the B cells integrate. From dendritic cells and macrophages, neutrophils, eosinophils, NK cells, NKT cells, can all provide signals that might influence T and B cell behaviour. And as a consequence of all of this integration and all this signalling, B cells make decisions and T cells make decisions about what they'll become. They can become different forms of memory with different antibody types. Plasma cells, short or long-lived. T cells can be different types from Treg, Th9, Th17, very diverse. And of course we know now from single-cell transcriptomics as well that if we drill in keep drilling down to look at population-level behaviour, again, just as we talked about with the receptors, expression of transcripts within individual cells is also highly diverse, and any single cell will have its own unique signature, which has more or less unknown contribution to the ultimate immune response. So this is the theme of my talk then. How does all this decision-making and fate regulation work? And can we find general principles of cellular calculation and control that might help us make progress on what looks like a very complex problem? So we're taking all these inputs into our cellular calculator and the outputs of how we affect division, death, differentiation and movement of the individual cells or the population. Okay. So the way, where I'm going to start here is looking at this canonical immune response. We activate the cells here, we see expansion, typical pattern. And if we look at, if we just think about T cells, what does it take? We ask, what does it take to get that response started? Now, if you look in the textbook, you might see that there's this is sort of a switch-like process. We have one signal, antigen alone might lead to tolerance. Second signals, like co-stimulation from CD28, or in addition to cytokines, are required to turn off the tolerance fate and turn on the activation fate. But of course the problem is, over the years, many different signals have been identified that will influence T cell proliferation. CD28 is quite good at it, but you can knock out CD28 and still get effective T cell responses in some situations. There's lots of cytokines, IL-2, IL-2, IL-12, IL-6 can influence T cell behavior. CD27, other ligands influence T cell behavior. So leaving us the challenge of what does each signal do and what is its relative importance in activating the T cell. And this is really where I start my story of experiments here, which is and I'm going to summarize quite a long journey of experiments trying to answer this question of how cells, T or B, integrate information and signals and try to convince you that in fact the rules they're following are really quite simple. And even though they're simple, the possibilities are incredibly heterogeneous and diverse. Okay. So we're going to start reductionists in vitro and try to eventually work towards in vivo experiments. So this is a paper just summarising the approach from quite a few years ago now, because the impetus for the experiments I'm going to describe was the discovery of division tracking dyes. C4C was the first. You could label up lymphocytes. They showed up brightly on the flow cytometer. Here's some peaks here. So here's a flow cytometry. This undivided peaks here after, 60 hours of T cell stimulation. Cells will, if they divide, they dilute the division tracking dye in half, and you can see that on the flow cytometer. They divide again, they have a quarter, and all the progeny have a quarter. Divide again, they have an eighth. So you can follow the progression of cell division by looking at the progression through these tracks by flow cytometry. And what was immediately apparent was what you see here, that even though all the cells are getting the same stimulus, they haven't all divided the same number of times at the same time. There's some heterogeneity there. And so we tried to track the source of that heterogeneity, and we discovered that we could describe it with just a simple model with just 3 parameters. What seemed to be happening is illustrated here. The first part of the model is that there's some variation in the time the cells in the population will take to enter their first division. We originally proposed that it was sort of a normal distribution. We've measured it more carefully now and know it's log normally distributed, or very close to log normally distributed here. And that it's these early cells here in the time to first division that are at the front of the division peaks. And it's these ones that take longer to get started that are at the back of these division peaks. But once they start, they tend to tick through divisions relatively synchronously and with much less time than they did to get started. And by plotting the average division number of the population here, so the average division number over time, from the slope of that line we can work out what that subsequent division is. So over here I've just used a normal distribution with a mean and a standard deviation here and a subsequent division time as here to recreate just in Excel the original data over here by fitting parameters. So just 3 parameters can recreate this behaviour. And that allowed us to ask, what did signals do if we add in a co-stimulator in here? This is all done just with anti-CD3 in the saturating presence of IL-2. If we add in anti-CD28 and ask, well, sorry, I'm getting ahead of myself. We can ask, do signals influence the time to first division? Do they influence the standard deviation of the first division? Or do they influence the time through subsequent division by changing the slope here? So this is a summary of lots of experiments. So this is anti-CD28 moving the time to first division but not the time through subsequent division. IL-4 moving both a little bit. The combination summing together in a sort of arithmetic way as if they're independent contributions to the behaviour. And building the little model, those small changes, which are 37% changes in time to first division and a near 10% change in the time to subsequent division, small changes are sufficient to give this big difference in cell number after 3 or 4 days in culture. So the conclusion for the single cells were highly variable but collectively predictable by describing the main source of variation as a probability distribution. That death and division seemed both stochastic following distributions, but they appeared to be independent of each other. Multiple signals added independently, allowing the net effect to be predicted, and multiple small differences magnified in this sort of nonlinear manner. Now, we've gone ahead and looked at this sort of problem in many more sophisticated ways. That's not my theme today, because I want to introduce you to the next part of the immune response, which is that it stops. Cells divide, and then they will stop. And the question is why and how is that influenced by the combinations of signals? And how can we fit that together? And it turned out that a clue here was from filming B cells. So B cells, when they're stimulated with CpG, unlike with CD40 that we heard about before, they don't aggregate together. They grow nicely and separately, so you can follow every individual cell and build up complex family trees of their behaviour. And a PhD student of mine, Edward Hawkins, set this up in the lab and followed hundreds of different families and noticed that this is cell size versus time here. They would grow, divide, grow, divide, grow, divide, and eventually they would die. But you can see in this last generation, after their last division, there's no growth. And you can see also that they're getting a little bit smaller every generation. There's a very consistent pattern. The other interesting point to note was that within the cells, and we looked at hundreds of cells, the numbers of times they would divide varied in the population and followed a distribution. Divide once, twice, 3 times, 4 times, 5 times. But if you look within any individual family, it turned out that almost always we saw patterns like this. So all the cells of a single founder would divide the same number of times. They would stop dividing in the same generation and go on to die in that same generation. So this variation is at the population level, but in any individual family, the information for how many times to divide is programmed into that first cell. So we call this the cell's division destiny. How many times will it divide before it stops? Okay. Now we could build fully probabilistic models of these processes. Variations in time to first division, variations in subsequent times to subsequent division, the number of times cells divide before they stop, and the times to die, if they stop dividing, the time they'll take to die. So we could build that model, fit it to data, this is CpG data, that works beautifully. And I just mentioned that because I'm going to use this model a little bit later to fit some T cell data. We call this the Cyton model and it assumes independence of control of division and death. So, going back to T cells, we didn't see that proliferation stop and die behavior in T cells. But usually our T cell cultures, we reasoned, were stimulated with antigen-presenting cells, which would have lots of co-stimulators, or they make IL-2, which can provide potential maintenance of the proliferation. So we thought, well, maybe what's happening is here, if we could strip the T cell activation away, pare it down to a bare minimum, maybe we'll see this autonomous behavior where we activate a sort of mini immune response where cells will divide a number of times, stop, return to quiescent, a small cell, and eventually go on to die, just like a mini immune response. Somehow that complete behavior is already programmed into that first cell in the B cell. Could we see evidence for that in the T cell? So what we did, and this is the work of principally, in this case, a very talented PhD student, Julia Marchingo, in the lab, working with a postdoc in the lab, Sue Heinzel. So here's the system she set up. We would take T cells, these are from a transgenic T cell specific for a peptide, the CD8s. Wow. That's good. Okay, so we take these T cells. If you label them with, in this case, cell trace violet, so we can watch the cells dividing. We're stimulating with peptide alone and no APC, so they're self-presenting to minimize the amount of co-stimulation they're receiving. We're also blocking all the IL-2 with an anti-mouse IL-2 antibody. I'll show you later, we'll come back and look at the effects of IL-2 by adding in human IL-2, which is not blocked by this antibody, so we could buffer the level and analyze the cells by FACS. And you can see when you do that now and you stimulate the cells, they indeed divide a few times. By 65 hours, you can see we've got some undivided cells here, once, twice, 3 times, 4 times here, 5. And then they just stop and start dying, just as the B cells had done. So we've got, by minimizing the stimulation, providing that the cells were following the same behavior. And they also returned to being small cells, just like the B cells had done. Now, I'm just going to show you an example of this as a little movie, because we filmed these cells and it illustrates more than I can say by showing you lots of CTV plots. This is a Fookie movie, so these cells are reporting their cell cycle. They're red in G1, green in S phase G2M. This cell's just divided, and it's being imaged over about 2.5 days. And you can notice that it's crinkly. I think it gets better. So you can see it going S phase. What I want you to notice, so we've divided twice now, 3 times now. You can see the synchrony of this little clone. 4 times. And 5 times. They all divide 5 times. They all stop dividing and they all go on to die. Just as we saw in the B cell behavior. And we see this over and over and over in these clones. We filmed many, many, many cells in this way. So the same behavior we saw in B cells, we're seeing in T cells when we pare back the stimulus. Okay, so now I'm going to ask the question, how do you regulate the number of times T cells divide before they stop? What do all those different signals do to that process? And this is the assay. Filming is really tedious to do that, so we went back to flow cytometry. We can track cells moving through divisions, as shown here, stopping and starting to die. And if we just plot the mean division number versus the percent survival of, as a percent survival of the founder, so taking away the effect of division, the expansion. So if there's no death, the cells will move through generations and just not die like this. But you can see this behavior, they divide, they stop, and they start dying. And so this gives us a clue at the generation at which they reach. So we can sort of read it off the scale here. So we're going to go through and ask what affects how many times T cells divide before they stop. And you can increase the affinity. With OT1, you can play around with the affinity quite easily with the peptide, and that indeed affects the number of divisions. You can put in anti-CD28, titrate it in, and you can see that indeed affects, in a concentration-dependent manner, how many times the cells divide before they stop. Interestingly, that has to be seen before the first division. You can't add it later to influence that fate. The same is true for anti-CD27, another co-stimulator on the cell surface. Looking at cytokines, IL-2, not surprisingly, influences the number of times T cells divide before they stop. IL-12, IL-4, IL-6 a little bit, IL-21 a little bit, and just as negative controls, IL-10, interferon, nothing.
So all of these are before the first division?
What's that?
All
of these are in constantly at this.
But the ones above are before the first division.
In this experiment, they're there for the whole time, but we asked when were they necessary. So if we take the anti-CD28 away after 24 hours, we still see the same behavior. And if we add it in after 24 hours, it has no effect. That's not true of the cytokines. That's acting both before and during. Now, just to convince you that this is autonomous, we did this experiment, cute experiment, where we stimulated the T cells with a weak stimulus, so just the peptide only, which is just enough to get them to divide about once and then stop on average. We can put the peptide in with a combination of CD28 and IL-12 here, which is a strong stimulus. That'll get the cells to divide about 3 or 4 times. Okay. And so we can either culture them alone and follow the proliferation and stopping, or we can culture them together in a V-bottom well, so they're all mixed together. But because we've labeled them differently, we can identify them by facts. And when we do that, you can see that the proliferation, cessation, and death of the two populations, when either cultured alone or together, is the same. So it's sort of an autonomous programming of their proliferation fate. Okay. So now we can ask, how do the signals add together? Play this game. So is there a quantitative rule for adding multiple signals? Now, let's add just peptide alone will give us about an average of 1 division here. We can add a low concentration of CD28 to give us another division. And we've got to keep it low because our CTV, we can only track up to about 6 or 7 divisions. Okay. So CD28 is giving us an extra division here. CD27 is giving us about another extra division here. If we add them together, okay, we can measure the outcome. Now, I am going to do this. So I'm going to ask you to predict. So you can predict it simply adding the 2 arrows here, this number and this number, would give us this dashed line, the additive effect. You might imagine that one would be dominant and it would fall here. You might imagine some synergy and you'd get more than the proliferation of either alone. So who's going for dominant? I'm not going on until you've all voted. So no one wants dominant? Who's going for additive? About half. Who's going for synergistic? About half. Very good, thank you. Okay, there's the answer. Additive. So pat yourself on the back, either because you guessed or you read the paper. Well, that's less good. Okay. Okay, I should say Julia did many combinations, 2 and 3 of different stimuli, and every time it came out in this very simple manner as additive. Oh. And I should make the point too here that of course we're adding together on a division scale. So every time you add a division, you're getting a 2-fold expansion in the cell number. So if you add 2 divisions, you've got a 4-fold expansion. And so we're sort of integrating this linear manner, but we're getting a geometric increase in the immune response outcome. So that's an important point, I think. Okay, so can we calculate the addition of signals in vivo? Julia did this beautiful experiment, which was a lot of work, where we played with 2 variables. So you can transfer into a mouse the OT1 T cell and infect the mouse with a version of influenza that expresses the peptide that the T cell sees. And we have 2 versions of the flu. One expresses the wild type, the high affinity peptide, and another a low affinity version of that peptide. So this is Q4. Q4 is the low affinity version, N4 is the high affinity version. We can also take the OT1 T cells that have been crossed onto the IL-2 receptor knockout mouse. So this mouse, so the T cells from that mouse can't receive any IL-2 signals. So here's the experiment. So this is the OT1 response over time, following the mouse, pooling all the lymph nodes and looking at the proliferation, cessation and loss of cells. A typical immune response, log scale here, low affinity, no IL-2. If we allow those T cells to integrate IL-2 signal in vivo, we get a difference, we get a higher response here. And the lines that are fitted here are using that Cyton model that I showed you before, where we're predicting the number of times cells divide before they stop. So this is with low affinity, this is with the low affinity integrating IL-2. We have a difference induced by IL-2 here of this much. We can now use the high affinity peptide without IL-2 and predict this number of divisions before they stop. And we can look at the difference between low affinity and high affinity without IL-2. And now we can add the 2 arrows again in vivo for IL-2 and TCR affinity and predict this distribution, which is here, and the experimental data falls nicely on that curve. And even if there were a 10% difference in the destiny here, we would pick that up easily as a difference in the cell numbers there. Okay, so just to summarize a little bit here, what we're proposing is a replacement of this sort of 2-signal idea of T cell activation as sort of a switch with a continuum of signal integration. So illustrated here by some combination of signals that turn out to be very weak versus a combination of signals that turn out to be very strong, allowing us to play around with times to divide, times to die, and numbers of times you divide before you stop. So this is sort of color-coded, so they're sort of modelled here in a colour-coded way, and so we can take a system where we stimulate the cells very weakly, we get very little division and principally death, which looks like one signal tolerance. We can activate and get rapid proliferation that dominates over the death, and so we get rapid proliferation. Eventually they stop and start dying with this long tail log normal distribution. And the important point, everything in between is possible as a consequence of the integration of these signals. So, and I mentioned earlier, and of course we're all aware that there's T and B cells that are integrating lots of signals from other cells, numerous sensing signals that can be integrated, the idea in many different combinatorial ways. Many different ways, as Julia showed, many different combinations of signals could give you 10 divisions of T cells. There's no one particular activation path. And so we can think of this as sort of a quorum nonlinearity. All cells make some collective decision about how strongly we should respond to the threat that we're being exposed to. So everything, whether it's coming from an NK cell, a macrophage, some other source, is being integrated by the T cell to decide how strongly it should respond, how quickly and how many divisions it should undergo. So here's our illustrate T cell proliferation here, shown here, with different combinations of signals pushing it further and further. And the important point, at least in this conclusion, that many different combinations of signals here illustrated by numbers and letters could give you the same net outcome. So there's no single hardwired on-off mechanism for T cell activation here. Okay. I'm going to now ask the question, okay, I've shown you that both in T and the B cells there's this controlled division burst, division destiny programmed into that first cell. What is the mechanism? We thought we should be smart enough with all the modern tools to work that out. And this is principally the work I mentioned already of Sue Heinzel. And we started with a hypothesis. Here's the hypothesis. We thought what should happen is that within that T and the B cell, in response to the combinations of signals, something would be made inside that cell that would motivate division. And then with every division, you would dilute it in half, and then dilute it in half until you drop below a threshold, and that stopped the cells dividing. That would fit with the cells getting smaller every generation, perhaps. And we also knew from analysing our data that the bigger the cells got here, the more they divided as a clonal family. Okay. Now we could have done lots of proteomics, but we thought before we do all that, we'll just eliminate MYC as the possible counter. MYC as this molecule, and Mark's already introduced it for us. Important driver of proliferation, well known. We thought it should be something deregulated in tumours, because obviously if it went wrong, the cells would continue to divide. Tick. It should be a driver of proliferation cell cycle, tick, and it should it should be a target known to be induced by a T and B cell activation, and that's a tick as well. Okay, so MYC expression in dividing B and T cells. So this is B cells responding to CpG, and this is T cells responding to peptide and anti-CD28. We have proliferation, cessation, and loss, as we've just been talking about in both cases. And if we look at MYC, it had this satisfying pattern that we might have expected. It goes up and comes down, and it coming down sort of coincided with when we knew the cells were stopping to divide, and this was true in the T and the B cells. Now the important point here, though, is does it step its way through generations? And we can follow generations by our division tracking die. So when we do that, it turns out that MYC goes up, comes down, but you can see there's no change with generation. It's following a pattern, but it's not this pattern. It's actually this pattern. So the pattern of increase and loss, even though cells are unsynchronized and they're moving through division at different times, the pattern of loss of MYC is unaffected by the division. It's both in T and the B cells. So if MYC is what's responsible for this behavior, then it's behaving like a timer, not a counter. Okay. So that was a surprise. I haven't proved to you that it's MYC yet, but let's look at some of the other features. If we look at MYC level under different stimulations of different stimulation strengths, so weak, stronger, 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 and we know that as a consequence of these increasing strengths, the cells divide more and more, you can see the MYC level follows this satisfying increase in distribution. So it reaches a higher maximum and it starts declining at later times as you increase the stimulation strength. And using a little bit of quantitative analysis by plotting the average division number versus time here for the weak. So this is the plateau, shows us where they're stopping to divide, the weak to the strong. We can estimate the time at which each one, the average time in that population the cells are stopping to divide for the different stimulation strengths, shown by the dotted lines here in these colours. And if we take them down and overlay on a log plot now the MYC level in each case, you can see satisfyingly they all sort of stop dividing at the same low MYC level, consistent with this being a threshold that correlates with division destiny.
Could MYC be a readout for something else?
Yes. So then you have to ask, okay, I've shown you lots of nice correlation, it sort of makes a nice story. What's our evidence for causation? But why don't I just do that? Well, before I get to causation, I think this is important.
Don't let me, just keep going.
No, the other, well, okay. This point here is just saying not just stimulation strength, but if you add different signals, so CD28 or IL-2 in combinations, influences the level of MYC, and the level of MYC, as a consequence, correlates with the number of divisions the population goes. So it's not just strength, it's combinations.
This is a MYC protein, right?
This is MYC protein in the cell, yeah. Yeah, just intracellular staining. This is a very musical talk. I'm really enjoying it. I'm glad I put symphony in that. This slide I put in because it explains an important point. Why is it that MYC doesn't dilute with division? Or any protein? And this is probably true of many proteins. And the answer is that it's turning over incredibly rapidly. If we block all protein synthesis with cyclohexamide and follow the loss of MYC at any time after we've started the activation, it's about 20-25 minutes. Okay. So clearly what's happening is that the production rate of MYC is altering over time. And that production rate is being inherited through the generations. But what you're inheriting is the production and the degradation rate, not a particular level of MYC, which is, I think, really interesting. Okay, now we get to causation. And of course, to test causation, we have to force MYC in our population and show that they would continue to divide. And so with the help of Lynn Corcoran, she made for us a retrovirus that expresses MYC and it infects B cells very well. And we could follow the infected cells because they also express GFP. Now, over here, I hope I'm not putting you off by getting... So over here, so this is our cells proliferating. So this is just our typical CPG response. Cells start dividing, they stop, and they start dying. I've shown you that before. And now we've infected with the virus and forcing in MYC. And what happens is that, it looks like they're trying to divide, but then they all start dying just like the wild type, and at about the same time as wild type, and that becomes important. If we look at the average division number that the cells are undergoing, you can see they're trying to divide, they're continuing to go up even while they're dying here. So we thought, okay, MYC looks like it's pushing them into division, proving causation here. If it's there, they will continue to divide. But there's a death mechanism here, this famous apoptosis mechanism, that's overriding that attempt. And so we said, okay, if we could protect the cells from dying, which we could do because at the Walter and Eliza Hall Institute we have fantastic groups working on apoptosis. We had access to these BCL2 transgenic mice where the B cells take much longer to die. Okay, and so now when we infect those cells with the MYC virus, MYC expressing virus, now they just continue to divide until they eventually start dying much, much later. But you can see much more growth in the population now. And I'll come back to this point. So I think that proves that forced MYC expression in these cells will continue to maintain their proliferation. Okay, so I think it's reasonable to conclude that MYC here and the cessation of MYC is what's regulating division destiny in these cells. So let me wind up by telling you a simple model here that MYC looks like it's behaving as a temporal controller of division. So each cell is integrating signals, driving the production rate of MYC so it goes up proportional to the level of stimulation, and then follows a timer and it will stop dividing when it hits the threshold. The number of divisions you get through varies between cells because there's a separate timer for division. We can talk about that later. But in this particular cell, the division time is 40 hours and it stops dividing. Here it got through 4 divisions. So this looks like a cellular machine, a division-independent timer. Now I want to come back to death because when we look carefully at our data, we also saw evidence for a division-independent death timer. We could take this is T-cell proliferation, and slow it down with a drug, antimitotic drug, mycophenolic acid here. And what you can see in looking at mean division versus time, that they all stop dividing at still around about the same time, and they start dying at around about the same time. In fact, when we account for the amount of proliferation due to expansion of actual division and just look at the rate of death in the population here, in the unexposed to drug population here, it follows this curve, which is a log normal time to die distribution here, following. As we slow down the division here and here, just transposing the same distribution to here and here, you can see it fits the same curve. So the time to die is also followed in this population in a division-independent way, just as MYC is being passed through, or destiny time is being passed through generations, so too does it appear that the death time is being pursued. So we can build a model based on those ideas. 2 timers, effectively 3 timers because we put a division timer in there as well, but one controlling destiny and death here. Each cell follows a family distribution. They all follow the same fate, but within a population we have a distribution which we can measure for time for destiny and time to die here. And just those 3 simple machines in the cell account for the canonical T and B cell proliferation behavior. It can all be programmed into that early on in that cell, and it will follow a number of divisions. It will stop, return to quiescent, and go on to die, and give a beautiful distribution, as we see so often, following immune responses. So division only would have done this. Division plus division destiny would stop and not die, but the combination of all 3 gives us this nice, appealing behavior. Now we can use that to fit to data. So all that retrovirus data I showed you where we had controlled proliferation, where the cells divided and stopped and died, we put in MYC, they divided, they started dying even though they're trying to divide, or we can put it in BCL2 where they divide and don't die, or the combination with MYC. Put it on a log scale here, this is this data here, and we can fit that model that I just showed you where we have a destiny time and a death time in competition, and we've got the same proliferation rate for all of these set in the parameterization. It's a very simple model. These parameters can give this fit to the wild type, move the time to allow it for division by putting in MYC. We can get this fit, putting in just changing BCL2 and changing the time to die gives us this fit. So now the question is, if we put the 2 changes due to MYC and due to BCL2 in the model, can we recreate this rather remarkable behavior here? And yes, we do. So we have a 5 log increase here due to just simple 50% increase, 50% difference in the average time you're allowed division with this log normal distribution and average time allowed for death. Amazing sensitivity to small difference and the synergy of what's probably quite significant of 2 oncogenes here, going from this sort of highly regulated control to uncontrolled proliferation with just quite small differences here in the behavior. So insights for evolution of adaptive immunity. Both T and B cells seem to have programmable autonomous division bursts that are timed and not counted by MYC production and loss. It looks like it's indicative of some sort of primitive genesis. It's like being stolen from the organogenesis, proliferation, sort of autonomous proliferation, differentiation, cessation, and patterning. And that this simple autonomous response is regulated with many variations, as I've tried to explain. How am I going for time? I think I must be close. We're done.
How much Nick do you have left?
You have another 10, 15.
You sure?
Yeah, well,
Okay. Okay. Now we've been going back to all our filming data, which we looked at as if the cells were counting, but we've gone back and looked at it all now with the information that processes are timed and timed from first division and being passed through generations. And we've looked at the independence in particular families between the time the family took to start dividing, the time it stopped dividing, and the time the family started dying. And we find no correlation between those. In other words, the cell behaves as if it's got 3 internal controllers that are made independently and controlled independently, remarkably. And as a consequence of that independence, we can use this very simple model to capture this very complex behavior. Okay. Now, what is the source of the variation that we're seeing? You know, these lovely distributions. They're quite for each of those. And I started off by talking about making a perceptive cell with all those different receptors and different internal components, and they're all a little bit different. And so one hypothesis is that that contributes to this distribution. And just to give you an example of how this may be the case, when we look at T cells, Julia did this experiment again. When you look at T cells, after 24 hours, they have a nice log normally distributed CD28 population. And we can sort cells from CD28 low or CD28 high, put them in identical culture with N4 and anti-CD28, and follow their division destiny to see if that influences. And indeed, the cells sorted from CD28 high cells divide more than cells from the CD28 low, as an illustration of that variation in the construction of the cell contributing to some of these stochastic features. Okay, I'm quite near the end, but I just wanted to say a few words about differentiation. We've looked very hard at differentiation, principally in the B cell. Most of it's published that I could talk about. I'll summarize it a little bit because I want to show you one example, one new example that might be interesting to you, showing competition between fates. What we set up was with B cell differentiation. In vitro, we observed thousands of cells undergoing proliferation, switching to the antibody type from IgM to IgG, or becoming an antibody secreting cell. And we could follow this in little microwells where we're looking at a blimp reporter as a GFP reporter. We could look at switching by antibody staining, and we can follow it by visually division and death. So we looked at thousands of cells in this way. And just to summarize, the working with the brilliant probabilist Ken Duffy on analysing this data, we could show that in fact, even though it looked like, in fact, every cell looked different in what it exactly would do if you finally measured it against time and all the different possible fates, the entire complexity could be resolved by a competition model where 4 fates, division, death, differentiation, and isotype switching, were each following timers, each distributed in a log normal manner in our starting population. We watch them divide and follow them through. Principles of competition and censorship allowed very subtle correlation structures to be observed that were strongly suggestive of this model of independent behaviours of each of the fates. So in other words, 4 simple machines set up in competition automatically gives rise to this incredible heterogeneity.
Can you explain what you mean by they're set up in competition? They seem to be independent. At least some of them. Like
Yeah,
this,
so
switch.
let's say we make a cell
Yeah.
and we build in its differentiation machine. This cell will now have a timer that says, I will differentiate at 60 hours. There's a division machine that says, I will divide in 30 hours. They proceed along not knowing what each other are doing. If you divide first, you never see the differentiation. But if the differentiation timer proceeded along and in that cell it triggered at, say, 20 hours before the division, you see it. So they're in competition.
What you need is that one actually cancels the other.
That's
So
the censorship.
The first one
Yeah.
that happens
In this
actually
case,
then resets the timers for all the rest of them? Is that the idea?
division in our model, division resets them. It's the only thing. If you differentiate, you don't affect any of the other timers, except for switching. You can't switch if you differentiate. So there's little tiny.
Death is obvious.
Yeah, sorry.
What death does.
Death is the ultimate censorship.
I was wondering
Yeah.
about differentiation
Yeah.
isotype switch, because you said you had 4 different machines all in competition with each other.
Yeah.
That's not exactly what you mean, I don't think.
Yeah, plus rules of censorship. Okay, and the rules of censorship are division resets everything, so you redraw, you remake your machines. Death, it's all over. Differentiation, you don't affect death or division timers, but you turn off this. And we know from our experiments, you've probably done similar experiments, if you become a plasma blast, you can't switch any further. If you switch, you do nothing. There's no censorship. You just proceed.
Okay,
So-
they're all related to each other in different ways.
Yes. Small ways. Okay. Sorry.
You know the Steve Reiner work where you get the asymmetric cell division that partitions the components within the cell
Mm.
to force them to go to a vector or to memory. And you probably thought about that. Or not.
You're
You don't
asking.
believe in it?
Well, all I can say is, we're looking at B cells. We're looking at
He has it for B cells and T cells.
yes. All I can say is, in all of our imaging, there's unbelievably high concordance between the fate of 2 sibling cells. Like, it's remarkable. And where it's not, using our model, we predict the level of asymmetry as a consequence of the competition. So all I can say is we don't see it. We don't need it to explain, infinite level of heterogeneity. However, we don't do exactly what Steve does. So I'm not going to say he doesn't.
That's what
Yeah.
I mean,
Yeah.
to add to that discussion, Mark, he only ever showed it in B cells, supposedly in vivo with germinal center cells.
That's right.
It's based
B
on like
cells is
3
just
cells.
one. The T cells is center lot.
Well, CD8 T cells he has, yeah.
Yeah.
But it's based on like 3 or 4 cells. I mean, most of us have looked this over. And
He's not the only one.
several of us have tried to reproduce that and not. You can't reproduce something because you could have done it wrong, but I think it's in considerable doubt. And if you look at the preponderance of evidence in careful studies from Phil, I just don't see how those 2 biologies can really go together. And you have this,
Yeah.
like you said, thousands of cells under multiple
Yeah.
conditions there.
Other people are very palm.
There's a few.
On terminal center cells?
No, in T cells.
Yeah.
In T cells.
Not what he's talking about.
Yeah. I think that would be a good topic. I mean, it is a fascinating question,
No, it's a great
and we
concept.
have.
It's a fantastic question.
I can say, Edwin.
Could you repeat the question?
We're being asked about asymmetric cell division. So the idea that early on there would be an asymmetry in the fate of the 2 daughter cells, where one would pursue, for example, an effector class and another a memory class. And
You're
there's
saying that you don't.
I'm saying in our in vitro filming, the concordance between 2 siblings is extraordinary, which actually is an important other point I haven't emphasized, which is that this idea of stochasticity versus determinism, and I've shown you hopefully a lot of evidence for stochasticity, lots of nice distributions. But where does that come from? Because if you look at 2 daughter cells, it turns out their fate is very similar. So if you're a molecular clone, your fate is highly determined. So I would argue, I think what's telling us, and this would be a great subject for discussion for the week, is where that stochasticity comes from. Often comes from the mother in making the two daughter cells. Like it's randomizing something into its next generation. But having done that, the two daughter cells have the same determined fates. Amazing. So, okay, just to finish up, I just wanted to say something new for those of you who have heard my talk before. A bit of filming. This is Jay Zoe, who did all the original counting of those thousands of cells. She's been doing a PhD counting more cells. And one of the things we noticed was with CD40 stimulation of B cells in vitro, as we lower the concentration of CD40, we get weaker and weaker proliferation. But interestingly, if we look per generation at the number of cells that have become plasmablasts, antibody-secreting cells, it actually increases with the weaker stimulation. So it's sort of paradoxical. We get more differentiation per division with weaker stimulation than with strong stimulation. And so we argue, well, that could be because the CD40 somehow is reciprocally related, the probability of differentiating is reciprocally related to the concentration of CD40. The division times stay the same and the time to differentiate gets shorter and shorter with lower levels. Or maybe the probability of differentiating just stays the same and the division times are changing. And so Jay filmed that scenario and was able to show, I think, really quite beautifully that the last explanation is correct, that the division times are changing. This is Kaplan-Meier cumulative distribution functions here, so they're uncensored already for death and differentiation. Death and differentiation times stay the same, but the division times are changing, and we can fit log normal distributions to these division times to explain these curves for each of these different fates. And that fully accounts for this strange phenomenon. In other words, the slower the cells divide, the more opportunity they have to differentiate. We get more differentiation, not because we're regulating differentiation, but because we're regulating division, and because they're in competition, it allows us to see more differentiation. Okay, to conclude, immune cells are built in multiple stochastic ways with independent components where actually, if you look really hard, every cell is different. The properties of immune cells arise from the ensemble, and knowing these distributions of various functional modules that you're likely to encounter and how they're affected by the signals, you can deduce the overall properties of the whole. You can use these sort of statistical models which don't need molecular information and don't need to know the randomizing source either. Of course, it would be nice to know those, and I hope the mix story gives an example of how the mechanism, looking at the mechanism, can inspire how to look at the problem. Key fate control arises apparently from competition and censorship, accounting for significant population heterogeneity. We've got infinite variety, easily coded, and just to bring it back to the two steps, the stochastic generation of the repertoire, and I would argue that there's an enormous contribution of stochasticity in generating the second repertoire, all the cellular heterogeneity as a consequence of these processes. I need to thank very much Julia and Sue, who did the bulk of the work I showed. That last little bit was Jay, and of course all the probabilistic modelling and brilliance often comes from my collaborator in Ireland, Ken Duffy. Thank you.
Let's rapidly have a few questions, and then we will go. The microphone. Where's the microphone? Okay. So yes.
So if all you care on Earth is a weighted average of a lot of different signals, why do you need different signals? I mean, one signal is enough to provide the information of the average, right? You have 10 different signals, and you sum them precisely, like in a precise additive way. So actually, you only need one signal, or 4.
If I understand your question,
So you have like 20 different cytokines. And what you do, you actually sum the effect of all the cytokines. So that would be the same thing as just having one cytokine where you tune the
there's no
level.
doubt that what cytokines tend to do is manipulate a probability distribution that may be there already. So it can stretch it out, it can move it, And the net effect can be quite striking. So it can have a huge impact on the total number just because, you know, cells died less or they're dividing a little bit quicker. Similarly, it might stretch out the distribution, so you get cells spread across different generations. And we think, as I showed with, maybe I didn't, isotype switching at least is one fate that is very dependent on moving through generations. So the way you spread across the division profile affects your fate. And so by signals can affect the combination or the population diversity by playing around with these signals in quite, you know, remarkably striking ways. Maybe I haven't, you're looking at me as if I haven't answered your question.
Yeah. So suppose you have 10 different signals, and all you do is just to average them.
Oh, no, you're not averaging them though, because what happens is, so for example, IL-12 will principally affect cell survival and have little effect on division. In B-cell example, TGF-beta, for example, fantastic, highly anti-proliferative. It slows down and stops the bulk of cells dividing, but of those that divide, they very rapidly switch to IgA. So you can have these sort of paradoxical combinations of effects that are where the cytokine can have independent effects on different fates within the cell. So, and then when you mix those cytokines together, you're getting paradoxical effects. Then you really have to do that by experiment. It's hard to predict what it would be.
So how general do you think the additivity is? I mean, could you find synergism? Say if you take two molecules like CD8 and the T-cell receptor, would they show some synergism? What do you think?
That's an interesting one because CD8, in a situation where the signal on its own may not do anything. Yeah. Well, I guess the CD28 is a little bit like that. It doesn't do anything on its own. It just adds. And don't know. Otherwise, other than to say, you know, make the point again that we were adding together linearly, but it was, if you looked at it a different way, it is synergistic because you're getting more than the sum in the actual cell number. So you can look at it one way and it's arithmetic, and another way it's synergistic, depending on how you.
So let's thank Phil one last time.
Oh.